Okay, so this is the third lecture out of chapter 3. So in this lecture, we're going to talk about this concept of mass percentages, empirical formulas, and molecular formulas. So mass percentages, it's a pretty easy concept, probably one of the easier part of this chapter. So if we take an example, so first of all, what's the name of this compound? You should be able to name that hopefully by now. It's chapter 2 material and you're keeping up with me in class, right? So you already got all of this chapter 2 stuff down, hopefully. So Cu is copper. And PO4 is, if you remember PO4, first of all, what's the charge? It's 3 negative. That is phosphate. And copper is a transition metal, which means you need Roman numerals in here to indicate what the charge on the metal is. So what's the charge on the metal? So phosphate's -3, and there's 2 of them, so that's 6 negatives. So copper has to balance that to 0, but there's 3 coppers, so each one is +2. That gives you 6 positives, and to balance the 6 negatives from the 2 phosphates, right? So that would be copper 2 phosphate. Okay, so what is the mass percentage? Sorry. I changed the problem, but forgot to change all of that. So what is the mass percentage of copper, phosphorus, and oxygen in the compound? So if you want to do that, then first you need to calculate the molar mass of the compound. So going to your periodic table, so copper is 53.546 grams per mole, and there's three coppers, so multiply that by 3. Phosphorus, 30.9738 from the periodic table. How many phosphorus are there? There's 2, right? Because that subscript 2 extends to the phosphorus and it extends to the oxygen inside the parentheses. So how many oxygens are there? So take the molar mass of oxygen times 8, right? And so if you add all of that up, you should get 380.58 grams of copper 2 phosphate per 1 mole of copper 2 phosphate. So now we can calculate mass percentages. So mass percentage of copper, for example, is just what is the total mass of copper divided by the total mass of the compound, and then multiply that by 100%. So the total mass of copper is right here. It's the 53.546 times 3 divided by the total, which is right here. The total mass is right there. 380.58 times 100%. So if you do that, okay, let me calculate these in a minute. So phosphorus, so 30.9738 times 2 is the total amount of phosphorus's mass divided by the total, 380.58 times 100%. And then oxygen is 15.9994 times the 8 oxygens, right, divided by 380.58 times 100%. And I don't have to calculate it because I have it right here. Haha. So it's 50.09% copper by mass, and it's 16.28% phosphorus by mass, and it's 33.63% oxygen by mass. And of course, if you add these 3 numbers up, you better get 100%. If you don't, you made a mistake somewhere, and I haven't checked. This is a good way to check yourself. So let's add those up, and I hope it adds up to 100. Otherwise, I'm going to have to pause this video and start over. So yes, it adds up to 100%, right? Okay, so if you have, so that's how you calculate mass percentages. Just calculate the molar mass, and then for each individual atom, total up its mass and divide it by the total of the entire molecule. Okay, so if you had a 25.2 gram sample of copper 2 phosphate, how much of that 22.5 grams is copper? How much is phosphorus? How much is oxygen? It's a pretty easy calculation. So you would have to calculate the mass percentages first. So we've got the mass percentages now, so we can convert that to a fraction. So just divide by 100, right, to get rid of the percentage and convert it back to decimal. So how much of this is copper? So we would take the 25.2 grams in the total sample and divide it by the fraction. And that's how much would be copper. So 0.5009 times 25.2. So there's 12.6 grams of copper in that 25.2 grams. So do the same for phosphorus and oxygen. So take the how much you have times the fraction. 25.2 times so 4.10 grams of phosphorus. And then the same for oxygen. Take what you started with, 25.2 grams times the 0.3363. So that would be 8.47 grams of oxygen. And so there's a good way to check yourself. Make sure you didn't make any mistakes. How would you do that? So just take that number, and that number, and add it up, and you better get 25.2 back. Otherwise, you made a mistake somewhere. Whoa. I made a mistake in my calculator, I think. So yeah, you get 25.17 grams. Why isn't it exactly 25.2? Because of rounding errors, right? If we didn't round these numbers at all, then we would get the 25.2 back. Okay. All right, so let's take another example of showing you how this chemistry can be applied. So let's say you're a geologist, a business person. You have two properties, one rich in FeO and one rich in Fe2O3. So if it costs the same to mine each and it costs the same to process the dirt and rocks from the mining operation from each, and the same expense to isolate the iron from the FeO and the Fe2O3, so which would be more profitable to mine? The site with the FeO or the site with the Fe2O3? And so we'll assume each ton of dirt or rocks has the same mass of FeO and Fe2O3 in them. Okay, so FeO is known as woostite, Fe2O3 is known as hematite. So that's the common trivial name for them. So what would be a technical name? So what's the technical name or scientific name for FeO? Well, Fe iron is a transition metal, right? So you need to know what the charge on it is because it's going to have Roman numerals in the name. So how do you know what the charge on the metal is? Because you know oxygen. How do you know oxygen? Because you memorized it. Oxygen is always a -2, right? So if there's one iron and one oxygen, then they both, if oxygen's -2, iron's +2, so this would be iron 2 oxide. And here oxygen's -2, there's 3 of them, so that's 6 negatives. And there's 2 irons to balance those 6 negatives to 0, so each iron must be +3 to give you 6 positives, right? So this would be iron 3 oxide. So if you just tell somebody you need some iron oxide, nobody knows what you're talking about. Are you talking about FeO or Fe2O3, or there's actually other oxides of iron as well? So you have to have that Roman numeral in there so that a chemist or anybody knows what you're talking about. Okay, so how are we going to figure out which is more profitable to mine? We're going to calculate mass percentages. So use your periodic table, look up iron, look up oxygen. So 55.847, and there's 1 iron and iron 2 oxide, and there's 1 oxygen. So if you add that up, you get 71.8464 grams per mole. And for iron, I said 3 oxide, hematite, then there's 2 irons and 3 oxygens. So if you multiply that out and add them up, you get 159.6922 grams per mole. So now we can calculate mass percentage of iron. So we're going to take the 55.847 and divide it by the total, 71.8464. Let me get my notes. So times 100%. and that would give you 77.73% iron by mass. And so since there's only one other element present, oxygen, then the rest of the percentage is oxygen, right? And then for the iron 3 oxide, so the 55.847 times 2 divided by the total, 159.6922 times 100%, and that would give you 69.94% iron by mass. Okay, so if you had a kilogram, 1,000 grams of iron 2 oxide, how much iron is there? Then that would be 1,000 grams times 0.7773, right? So that would be 777.3 grams of iron. And if you had 1,000 grams, a kilogram of iron 3 oxide, then multiply it by the fraction that's iron, so 699.4 grams of iron. So obviously, on a mass per mass basis, you get more iron out of iron 2 oxide than you do out of iron 3 oxide. So if it costs the same to mine both of them, then it would be more profitable to mine iron 2 oxide because you get more iron out of it. So FeO would be more profitable to mine under these circumstances. In reality, it's probably not the same cost to mine the two, right? So the actual business model would be much more complex than this. So why would one want to mine for iron? Because you can build things with it, right? Steel is about 98 to 99% iron. It's mixed with a little bit of carbon to change the properties a little bit. So obviously iron is a very important element that gets mined on a huge scale. It's the 4th most abundant element in Earth's crust. Okay, so empirical formulas and molecular formulas. So first of all, what are they? So empirical formula just shows the smallest ratio of elements on a molecule. And we'll work some examples, so hopefully it becomes crystal clear what that means. Molecular formula, in contrast, shows the exact number of each element in a molecule. So I've given you some molecular formulas here. So C6H4. So whatever this molecule is, it has 6 carbons and 14 hydrogens. So its empirical formula would be the smallest ratio. So if you want to get the empirical formula, you find something that you can divide both numbers by to get to the smallest possible ratio. So you could reduce this to divide both by 2, so 3C3H7, right, would be the empirical formula for C6H14. If you wanted to reduce C6H6 to its smallest ratio, well, that would be a 1-to-1 ratio, right? So CH would be the empirical formula there. Or C6H12, its empirical formula, so you divide everything by 6. All right, so you find the largest common divisor, and you would have C1H2O1. Of course, the 1s are typically not shown, so just CH2O would be the empirical formula there. Okay, so how about this one, C3H14O2? Well, there is nothing that you could reduce it to further, right? So in this case, so it is possible that empirical formula could be the same as a molecular formula if it's not possible to reduce it any further. Okay, so the significance of this, so if you have a compound's empirical formula, then you can determine its molecular formula, right? Why would somebody want to determine a molecular formula? Well, maybe it's a pharmaceutical agent, and if you want to know what the molecule is, then you need to know its molecular formula, and we'll talk about that in more detail in a minute. So you need to know, if you want to know the compound's molecular formula, you need to know the compound's molar mass. So how can you determine a compound's molar mass? So we talked about this back in chapter 2 when we talked about mass spectrometry and isotopes and how to determine an average atomic mass. So you need an instrument called a mass spectrometer, right? We have several mass spectrometers in the department. Cheap ones cost $20,000 to $30,000. The most expensive one cost about $400,000 to $500,000. Okay, so let me show you how you go the opposite way. So if you have an empirical formula, how do you determine the molecular formula? To do that, you need to know the compound's molar mass. So you would either have to be given that information, or you would have to be given a way to figure out a compound's molar mass, right? So in this case, I give you the compound's empirical formula, and I give you 3 compounds molar mass. So what is the molecular formula of these 3 compounds? So to figure this out, you need to find a multiplier to multiply the empirical formula subscripts by in order to get the molecular formula. So to do this, we're going to calculate first the empirical mass. So we have 1 carbon. Did I do this already? No. So for CH2O, the empirical mass, you calculate just like you do a molar mass. You look at the periodic table, and carbon is 12.011, and there's 1 carbon. And hydrogen's 1.00794 from the periodic table, and there's 2 hydrogens, and oxygen's 15.9994, right? And then you add those up. And I have this, or I just add it up. Let me just add it up. So that is 30.026, right? Okay, so then what we're going to do is we're going to divide the molar mass that's given divided by the empirical mass that we just calculated. So 60.053 divided by 30.026. So that gives you 2. So the actual molecule has a mass that is twice as much as the empirical formula. So how can it be twice as much? Well, you double all of the atoms. So we take this CH2O and we multiply everything by 2. We multiply the subscripts by 2. So C1H2O1 becomes C2H4O2, right? So if we did the same thing here, 180.16, the molar mass given divided by the empirical mass, 180.16 divided by 30.026 is 6. So whatever this molecule is, it weighs 6 times as much as the empirical formula. So the only way for that to be is for all of the atoms. So we take the subscripts and multiply them all by 6. So that would give us C6H12O6, right? And this one, the 150.13 divided by 30.026 is 5. So this would be C5H10O5, right? We multiply the 1 and the 2 and the 1 by 5. So that's how you can get from an empirical formula to a molecular formula. You have to get the molar mass, and you have to have the empirical mass, divide the 2 to find your multiplier, 2, 6, and 5, and then multiply all of the subscripts in the empirical formula by that multiplier. Okay, so what is this good for? So who cares, right? It's actually a very important part of chemistry, because if you can determine a compound's empirical formula, then you can use a mass spectrometer to get its molecular weight, and then you can use that information then to get the compound's molecular formula. And so how can you determine a compound's empirical formula? You get that from mass percentages. So let's look at how to do that. So how do we use mass percentages to get an empirical formula? So let's take, a mineral forsterite has the following composition. It's 34.55% magnesium, 19.96% silicon, and 45.48% oxygen. So let's use that to get the compound's empirical formula. So these mass percentages come from sending your sample off for what's called an elemental analysis. So chemistry departments can do that, private companies can do that, government labs can do that. They can take a sample, analyze it, and tell you the different mass percentages of all of the elements present. Okay, so let's use that to get an empirical formula. So to solve this problem, we're going to assume that we have 100 grams. Why 100 grams? Because percentages are based on 100%, right? So if we had 100 grams, then we must have 34.55 grams of magnesium, 19.96 grams of silicon, and 45.48 grams of oxygen. So once we've made that realization, now we have grams. So if we had a 100 gram sample, then that's how many grams of each of these would be present. We can now convert grams to moles. How do you convert grams to moles? Using molar mass. So you would need to calculate, So we can get the molar mass of magnesium, silicon, and oxygen just by looking at the periodic table, right? Okay, so we have 34.55 grams of magnesium. Looking at the periodic table, there's that many grams per mole. So 1 mole of magnesium is 24.3050 grams of magnesium. 1 mole of silicon, looking at the periodic table, is 28.0855 grams of silicon. And oxygen, 1 mole of oxygen is 15.9994 grams of oxygen, right? Okay, so we convert all of these to moles. So this would be 1.421 moles of magnesium. So in that 100 grams, there would be 1.421 moles of magnesium. And there would be 0.711 moles of silicon and 2.842 moles of oxygen. So now we're going to use those numbers we just calculated to write a formula, because formulas show molar ratios and we just determined the molar ratio, right? So we're going to use these numbers. So magnesium 1.421, silicon 0.711, oxygen 2.842. There's a formula. Now, of course, formulas always have whole numbers instead. So now that what we need to do is convert these numbers to whole numbers. So how do you do that? You're going to divide each of these numbers by the smallest number. So 0.711 is the smallest, so we'll divide each of these by the smallest. And when you do that, this comes out to be Mg2O silicon first, Si1, and O4. There's your formula. That's an empirical formula. Okay, so in this case, each number just happened to be converted to a whole number when we did this. It doesn't always work out to be the case, and we'll work in the next example will illustrate that. So if it doesn't work out to whole numbers when you do this, then there's one extra step that you have to do. Okay, so let's take an example to illustrate how this works and how this can be used for drug design, for drug discovery, so pharmaceutical agent discovery. So let's say that you're a scientist, a doctor on a ship before modern medicine. So everyone on board is sick. Their symptoms are weakness. They feel tired all the time, sore legs and sore arms. They have decreased red blood count. Since it's before modern medicine, I don't know how you would determine that, but we'll assume that you know that. Poor wound healing, so death can come from simple wounds and infections. So everybody on board is sick, and then your ship comes across this island in the middle of nowhere. Everybody gets off, and then eat some oranges, lemons, limes, grapefruits, so different citrus fruit, and then everyone's condition starts to improve. So you're a scientist, doctor, so naturally you're curious, right? So what questions would you start asking? Well, so there must be obviously something in the citrus fruit that is curing this ailment. But what is it? Where is it? Is it in the seeds? Is it in the pill? Is it in the pulp? So you're a scientist, so naturally you want to figure this out. right? So what you do is you take it to your lab and you isolate the different components in the seeds and the different components in the peel and the different components in the pulp, and then you feed that to the people on board to see if you get the same response, right? And so you do this, and then you find the component, which is a chemical, right, that works. So you find the component that works, but all that you know is where it came from. At this point, you have no idea what it is, right? But you're a scientist, so you want to figure out what it is. So how can you do that? So you have this compound pure. So what you can do now is send it off for elemental analysis. And elemental analysis can tell you the mass percentages in the compound. So whatever this compound is that's treating this disease or this ailment is 40.918% carbon, 4.578% hydrogen, and 54.506% oxygen. And now you have enough knowledge to know how to get an empirical formula from that, right? So you're going to assume there's 100 grams. So that means there's 40.918 grams of carbon, 4.578 grams of hydrogen, 54.506 grams of oxygen, and you're going to convert those grams to moles using the molar masses of each of these different elements, which you get from the periodic table, right? So 40.918 grams times 1 mole of carbon over 12.011 grams would give you how many moles of carbon? 3.4067 moles of carbon. And for hydrogen, that would give you 4.542 moles of hydrogen. And for moles of oxygen, that would give you 3.4067 moles of oxygen, right? Okay, so now we go to number 3. Now we write a formula using these moles we just calculated. And then we're going to divide each of these by the smallest. So divide each by 3.4067. And then that gives you this. So C1H1.33301. So in this case, it converted 2 of the numbers to whole numbers, but not all of them. It didn't convert the hydrogen to a whole number. So we have one extra step to do. So these are formulas. They need to be in whole numbers. So now what we need to do is find a multiplier that we can multiply 1.333, and 1 by to convert all of the numbers to whole numbers. So how do you do that? Well, you can solve problems the way that I usually solve problems. If it's not obvious to me, then I solve problems by trial and error. I just start trying stuff until I find something that works. So maybe you multiply all of these by 2. Maybe you multiply all of these by 3. Maybe you multiply all of these by 4. Just start trying stuff, see what works. So this would give you C2H2.666O2. Did that work? No, 2.666 is not close enough to 3 to call it 3. So let's multiply everything by 3. So that's C3H3.999. O3 is 3.999, close enough to 4 to call it 4? Yes. So this is C3H4O3. That would be your empirical formula. So we don't need to go any further. We don't need to try it times 4. So we just incrementally work our way up until we find whole numbers for everything. Okay, so there's our empirical formula. Now if we have a mass spectrometer on board, we can figure out the molar mass, and then we can figure out the molecular formula. So we take a sample of this, stick it on our mass spectrometer, and the mass spectrometer tells you this has a molar mass of 176.12 grams per mole, or 176.12 atomic mass units per 1 molecule. So now we can get the molecular formula, right? So we need to calculate an empirical mass. So I've done that for you here. So we take this formula, C3H4O3, calculate an empirical mass, it's 88.06, and then we divide the two, 176.12 divided by 88.06 gives us this multiplier 2. So we take our formula and multiply everything by 2, so C6H8O6, whatever this compound is, that's its molecular formula. So that's a lot of information, right? We've went from knowing absolutely nothing to knowing the compound's molecular formula, doing some very simple math here. But we did need two instruments along the way. We needed a way to do elemental analysis, and we needed a mass spectrometer. Okay, so as a scientist, are we done there yet? So are we done? No. So what would a scientist like to know? They wouldn't want to know the compound structure. Because if you know the compound structure, then you can find a way to synthesize it in lab. So for mass percentages, we figured out empirical formula. You will know how to do that in this class. And from empirical formula, so I expect you to know how to do that. And from empirical formula, you can get to molecular formula. And so I expect you to know how to do that. And once you have the molecular formula, then you can figure out the compound structure. But to do that, you need a lot of organic chemistry. So a lot of you, half of you will probably go into organic chemistry. So after you take organic chemistry, then you would know how to take the molecular formula and figure out the compound structure using things like NMR spectroscopy. So this is the compound structure. Right, that is C6H8O6. That molecule is ascorbic acid, so it's a pretty complex molecule. So why does a scientist want to know, or a chemist want to know, a molecule structure? Because if you know its structure, then you can figure out a way to synthesize it in lab. So then you don't have to land on islands and harvest fruit, right? You can just make the molecule in lab and administer it to a patient that way. So yeah, scientists would want to know how to synthesize it in lab. To do that, you need a lot of organic chemistry. This is an organic molecule. If you want to know how to make it, you got to take organic chemistry and study a lot of organic chemistry. What some scientists would want to know is they would want to determine the compound's mode of action. How does it work biologically? Its toxicology, its side effects. How is it metabolized in the body? So scientists that do that are called pharmacologists, and biochemists do that as well. So to do that, you need a lot of cell biology, a lot of biochemistry, some physiology, some toxicology, right? So this is basically how drugs are discovered. People go out into the environment and they isolate compounds, bring them back to lab, feed them to cancer cell lines, for example, and see if they kill cancer. And if they do, then you got to figure out what the compound is. And once you figure out the compound is, then you got to figure out a way to make it in lab. So does anybody know what this disease is that this ascorbic acid treats? So this ascorbic acid is also called, it's also called vitamin C, right? So what does it treat? It treats scurvy. So this disease is scurvy, which comes about from a lack of vitamin C in the diet. So it was proved in 1932 that this compound treats scurvy. So anyway, if this is something that seems interesting to you, maybe you want to be a natural product chemist. Maybe you want to go out into nature, scuba diving off the coast of Jamaica and bring some coral reef back to lab and see if there's anything in there that has antibiotic properties or antiviral properties or anti-cancer properties, for example. And then if it does, then you would, of course, want to figure out the compound structure. And then you would hand it off to a medicinal chemist, a medicinal chemist would figure out ways to make the molecule, or a natural product chemist would do that as well, but a medicinal chemist would figure out a way to improve on the molecule if it has side effects, and to figure out a way to synthesize it in very efficient yields. Or maybe you want to be a pharmacologist and study how molecules work in the body, right? And study their toxicity, how they're metabolized, and so forth. So if this is something you want to do, there's plenty of graduate programs that do this. UT Southwestern in Dallas, for example, has an entire graduate program that has a focus on developing new pharmaceutical agents. So you would get a bachelor's in chemistry or bachelor's in biology, for example, and then biology maybe go on into biochemistry or pharmacology, or chemistry go on into organic synthesis, for example. Okay, so just that's one more example of the significance of this. So this molecule is called Taxol. It's a very complex molecule, as you can see. So in the 1960s, the National Cancer Institute instituted a program, just like we talked about. They basically sent scientists out into the field, had them bring back samples to lab, and tested those samples to see if they could find any anti-cancer properties. And they came up with a hit. So this molecule comes from the Pacific yew tree. And it was found that there's a compound in the bark of the Pacific yew tree. So this was in 1967 that it was discovered in 1962 that it had anti-cancer properties. The actual compound was isolated 5 years later in 1967, and then it took another 4 years to figure out the structure of the molecule. And then it took 10 more years to figure out how to synthesize the molecule. So why synthesize the molecule? Well, this is an interesting molecule because it had a totally new way to kill cancer cells. Basically, it disrupted mitosis. The problem with it, the Pacific yew tree is an endangered species, and it takes 6 100-year-old trees to provide enough Taxol to treat one patient. So that's a problem, right? So to isolate the compound that comes from the bark of the Pacific yew tree, so what happens if you strip the bark off a tree? The tree dies, right? So you would have to basically kill 6 100-year-old endangered species trees just to isolate enough Taxol to treat one patient. So then this would be where synthetic organic chemists step in and figure out a way to synthesize this. So the synthesis, it's a complex molecule. Once you take organic chemistry, a structure like that would make total sense to you. But it took 10 years to synthesize. Is a 31-step synthesis from known starting materials. So one research group working on this synthesis, there are over 20 chemists working on this synthesis of this one molecule alone. So a lot of effort goes into developing new pharmaceutical agents. Okay, but anyway, we'll stop there. Hopefully you found some stuff that's interesting to you here. And, if you've had an interest in medicine, maybe you don't want to, or pharmacy, maybe you don't want to be a pharmacist or a doctor. Maybe you want to be a scientist and be the one that's discovering new treatments for diseases and discovering new medications, right? This is one thing that scientists do, one thing that organic chemists do, and people with biology degrees who go into cell biology and pharmacology do. Okay, we'll stop there.